Now, how was it that each codon was assigned to a different amino acid? This was the work of Marshall Nirenberg and Matthaei. And it was done in 1961, so not a very long time ago. Does anybody know what they did to figure this one out? Actually, I was listening to the story of this guy here, Matthaei. He was a postdoc, and he was given the job of figuring out the codons for the amino acids to figure out codons for the bases. So what happened is that they knew, he figured out there was this system, there was this in vitro system that he would use. It was a bacterial base, an E. coli based in vitro system, that if you would mash all this E. coli open and put this soup into a test tube and then keep it at 37 degrees, he would see, also he was using radioactive amino acids. So he would see that after a certain time, he would get radioactive protein coming out of that, meaning that that soup out of this bacteria was functional. It could translate whatever RNA was there into proteins. Now, he was trying to figure out what would happen if you would add different radioactively labeled amino acids, and what would happen to the whole system if you would, or how long the system would still last. So he had seen that after about half an hour, he wouldn't get any more protein. So he thought maybe the system is all dead. Maybe all these proteins are degrading and it just doesn't work after half an hour of incubation at 37 degrees. But then that was not the case, because he had this great idea of adding exogenous messenger RNA into that system to see if the system would still work. He isolated messenger RNA, he put in there, and like magic, he got radioactive protein coming out of there. So he says, well, the system is not dead, so what do you think is happening that he did not get protein after half an hour of the first round? I mean, think about what he has in there. He does not have DNA. He has all of the translation machinery in place. He has this in vitro translation system out of this bacteria. So why is it that he's not getting any more protein? Okay, what is one of the differences between DNA and RNA? As RNA is single-stranded and DNA is double-stranded. DNA is protected by a lot of proteins, and RNA has to be protected by something in order to be more stable. So what happened to RNA after a while? If you want to isolate RNA in the lab, is it easy to do?
No.
No, it's a pain. It's a pain. RNA is very unstable. So this guy, being very clever, he figured out that his system was okay, it's just that the RNA was decaying. After half an hour, all the RNA was gone. This is why he was not getting any more protein out of his system. So then he had this brilliant idea. Well, he got a nice job in Germany, and this other guy here, Nirenberg, got a nice job at Berkeley in California. And he was trying to crank up the code and figure out what the codon is for every amino acid. And he was forgetting about this other guy, Matthaei. But then Matthaei, being German, in a very methodic and very organized type of work, he figured out that he could prepare 20 different tubes with one amino acid that would be labeled in radioactive materials. So he will get, for each tube, he had radioactive alanine. And then for his second tube, he had radioactive tryptophan. And then for his third tube, he had phenylalanine. Then for the fourth tube, he had, methionine, okay? And so forth. So now is this guy with a very exciting project, 20 different tubes, one radioactive amino acid per tube. And now he wants to figure out how is it that he's going to figure out this codon business. So he had a whole chain of messenger RNA that was made of only one nucleotide. In this case, all uracils. So he had all of this poly-U chain for his messenger RNA. He divided whatever he has here into the 20 different tubes. He dumped a little RNA into every one of these tubes, and he wanted to see where is it that he's going to get his protein made, and what is the protein going to be made of. he knows that since he has only one type of base here, he's betting that he's going to get only one amino acid in a long chain. He has each tube amino acid that is radioactive, and then he dumps his RNA here, there, every single tube gets his RNA, and then at the end of his translation, in vitro translation, he sees that only phenylalanine was coming out of his initial RNA that had a poly-U chain. So he said, well, you know, eureka, that UUU is the codon for phenylalanine. And then he got really excited and he says, okay, if I put another type of codon, if I put A, what type of amino acid is going to come out of there? Or if he put C, and T. And then he started to experiment. He started AUC, and what comes out of there? So he in vitro synthesized this long chain of messenger RNA that had one codon. He knew the codon was 3 letters, so he said, for example, AUC, synthesize all that, putting every one of these 20 tubes with each label amino acid, radioactive amino acid, translate it, and then he filter it and measure the radioactivity and see which amino acid he has there. And he did this for all of the 20 amino acids. So imagine 20 times 20, that's one experiment. That's a lot of tubes. He must have a lot of motivation, and I hope he was at least paid a good salary. So here's the other description of his experiments. He has all these tubes. He puts the messenger RNA, and one of them is going to come up with his chain of polypeptide of only one amino acid. So this is how they figure out the codons for each of the amino acids here. How is it that the amino acids are put together? How is it that the translation is happening, or the translation is getting put together? They knew that they needed this adapter molecule that is also made of RNA. This is the tRNA, which I'm sure you had in another basic biology class. tRNA looks a little bit like this type of clover leaf structure. It has 3 loops, and then in one side you have the amino acid binding site. And then at the bottom, at the second loop here, at the bottom, you have an anticodon sequence. And then this anticodon is going to recognize the codon that is on the messenger RNA. We're going to go into a lot of details of this process here. Okay, I just put a step-by-step description of the experiment of what Matthaei did. He got these nucleotides. He has a poly-U polymer for his amino acid, for his messenger RNA, and then he puts into this in vitro translation system, warms up at 37, get the polypeptide chain, filters out all of the amino acids that did not bind into a polypeptide, get his one in the filter that is a polypeptide. Actually, that's why it did not go through the filter. It's a big polypeptide there. And then he checks out which amino acid that was. So now coming into the genetic code, I'm adding these slides so you know in the chapter exactly what part of the chapter we are focusing on. Okay, so now we go to the properties of the genetic code. Which one codes for each amino acid? We have here 2 major regions on the transfer RNAs. You have this acceptor stem that is all the way up there. That is acceptor, you can imagine, is accepting the amino acid. Everything starts with A, acceptor, accepting amino acid. You also have the anticodon loop that is at the bottom. And you know that each amino acid that gets accepted into this tRNA, it has to be a very specific type of reaction. So you know that when you have an enzymatic reaction, you can imagine, first of all, to get this amino acid stuck to this piece of RNA, you must have some type of an enzymatic reaction. So there must be some type of an enzyme that will do that job. And since you have 20 different amino acids, you must have at least 20 different types of amino acid transferases that will add the amino acid to this RNA. Now, how do you think the amino acid transferase will recognize this tRNA in order to add the amino acid there? You see, at this very tip of this acceptor loop, all of these bases here are the same in all of the tRNAs. So how is it that the amino acid transferase will get in there and recognize which amino acid to put in there? Well, it's not quite a mystery. It has to read the anticodon, and it has to have a perfect match in order to run the reaction for adding this amino acid up there. Then it's very important that the amino acid addition is very, very specific at this acceptor loop. Talk about the anticodon loop. Okay. Now, of course, it's very important to know that this anticodon loop will bind to the complementary code, which is the code on the messenger RNA. Recap the experiment that was done. So now we're going to look at the codes. And we're talking about the redundancy or the degeneracy of the codes. So we know that a particular codon can have Wobble base that can be a different base, and still you're going to code for the same amino acid. I always give you a table. Okay, so you have to know how to read this. Which one is the first base, which is going to be here, and then on the second base is going to be there, and then on the third base is going to be here. You have to know how to get them together. Okay, so here's going to be, for example, U is going to give you what?
Phenylalanine.
Phenylalanine right there. And then if you have U, C, A, it's going to be serine. So you just have to match them and figure out what is the answer for that particular codon. So going step by step, we have to know here that the first base is the most important. The first and the second bases are the most important for the codons. And the third base is the one that is more lenient, that you can have a bit of error. You have space for error there. So we also know that three of the codons do not code for anything. That you have here 3 of them, that is U, A, and U, A, G, and there's another stop codon here somewhere. Okay, U, G, A. Okay, so these 3 codons here do not code for anything. Those are stop codons. And you can imagine it's going to be, they will show up at the end of the protein. Now, what do you think happens if you have some mutation and these codons just show up in the middle of the protein? What do you think is going to happen? Right. It's not going to be making the protein you really want at the beginning, but it's still going to show up at the end of the protein, because what you get is a protein that is going to stop at that codon. So it's still at the end of the protein, but you just have a very small protein now. Okay, it's going to say stop, so you stop. The translation stops. And then another important thing to know here is that AUG is always the start codon, and that one is always the codon for methionine. In eukaryotes, there's methionine. In prokaryotes, is F-methionine. Okay, so this is just a visual interpretation of what the codon is. Here is you have your messenger RNA, and if you have a 3-letter codon, you have the tRNA, the transfer RNA, that is going to bring your amino acid, that's going to pair up with this codon. So the pairing of the first and the second are the more important ones, and then with the third one, you have a bit of leniency, it's a more relaxed binding. So here, you can see that the codon UCC will bind to anticodon AGG, but if you have a codon that is a UCU, you also bind to this anticodon AGG. This third base here is a more lenient type of binding. Okay, oh, here we go. Oh, sorry, got you the answer. Press a little too much. One of the practice type of questions. Through the Wobble, a single watt can pair to more than one watt. Big question. Anticodon can pair with more than one codon. The third codon on the Wobble base, that's what is talking about here.